Do you feel like maybe a greater urgency to fail faster? Because some things just aren't going to work. And you're going to, there's some trial and error in this process.
Oh, absolutely.
Yeah.
Absolutely. Fail fast, recover, and move on.
Yeah. Yeah.
Especially, now that we are looking at all sorts of biomarkers, right? So it's getting harder and harder to understand what these biomarkers mean. But if you don't see anything coming out early on, just move on, get what you can and move on.
Right.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe.
Hi there. Welcome to BioTalk Unzipped, where we unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin, life science professional from Kansas City. And today we are in Boston with the AAPS NBC conference. And alongside of me is the wonderful Binodh DeSilva, known for a number of years, Senior VP now of Bioanalysis over at Ultragenyx. Thanks so much for being here.
Yeah, thank you. Thank you for having me. It's fun.
Yeah. So in your role, you've been at the forefront of, pharmaceutical innovation for a couple of decades. And what drew you into this world of bioanalytical chemistry to begin with? What was the inspiration?
So my inspiration was actually my professor at Kansas. Yeah, so when I KU, so J-Box, right?
That's right.
So when I came to do my PhD in chemistry, he was an electrochemist by training. So, however, electrochemistry, and I didn't have a chemistry. And but this is almost 40 years ago. He said to all of us, not just me, to most of our graduate students, said that if you want to be an analytical chemist, you also need to go and get your experience in and background in a life science.
Yeah.
Because there's nothing in the future, there's not going to be anything called chemistry alone, pure chemistry alone. And that's where it led the journey. So then we were allowed to take courses everywhere, virology, biochemistry, molecular biology. So I got this breadth of education because coming from Sri Lanka, we were very focused on chemistry, physics, botany, zoo, right? That was the main game.
Okay.
But here, we were exposed to so many things at KU, and that's where I kind of got into the bioanalytical. I was combining pure analytical with biologics and all sorts of life science technologies. That's how I started.
That's great.
And then, started making antibodies and using antibodies as reagents for my PhD. And so it goes that back.
Goes back to then, and it springboarded you into this really fascinating career.
It is. And then, I owe a lot to Ron Boucher, Jean Lee, all those folks, right? For bringing us up. They gave us a chance when they were the behemoths of bioanalysis at those days.
Right. And that's kind of where I fell in love with this community a dozen or so years ago at the APA meetings and so forth, is that I had come most recently from a career in IT sales. Yeah. Before I got, before I was recruiting, scientists in biotech pharma, and it was dog eat dog world. It was, you're looking at your competition. What are they doing? How are they, what angles do we need to take? And then I come to this meeting where people from all these competitive, spaces, technically, your Pfizer, BMS, Merck's, all sorts of companies, and you're talking to each other just like, it was very collegial. It was like we're all in this academic environment and sharing, well, what do you do? Well, how do you do it? And what's the best methodology? And you've tweaked it this way. And so it's like everyone's getting together to help. How do we get these therapies to market? How do we help our parents, our kids, and our brothers and sisters who might need these medicines? Yeah. We talked to Mark Arnold last at PharmSci.
Right, sure.
And we were talking about some of the things that he had done.
Yeah.
And he's like, yeah, no, my dad's on this medicine that I helped.
Yeah, I know.
Like, oh, that's really cool.
It is.
And that's touching. That's
Yeah.
yeah. Good. So now you're leading bioanalysis at Ultragenyx. What unique scientific or strategic challenges come with developing these treatments in these specialized populations?
Yeah, that's a challenge that we have, right? Number one, our patient population is so small.
Right.
They're sometimes babies. They're very young. Very few adults, right? I mean, there are some diseases that have the longevity, and so we do have also adults and adolescents in our trials. And the challenges that we have are based on some of those demographics, right?
Sure.
We can't have the luxury of having a whole bunch of serum samples. Every sample is so precious for us,
Right.
and we need to take good care of it. So that's where the main challenge is, to make sure that we respect that sample. I mean, much more than anything, so that
Precious.
it's very precious.
My goodness.
And also to make sure that we use it appropriately for the appropriate assays, right? We can't run the whole gamut.
No, you have to be selective.
Be
Hopefully
selective.
make those really good
Right.
decisions.
So how do we kind of do the research to do the scientific investigation to understand understanding the disease pathology,
Sure.
and understand the disease progression and what is involved, what is the mechanism of action, and what are the ideal kind of biomarkers or whatever we are going to use to interrogate that disease. So that's a big challenge,
Yeah, I can imagine.
but it's so much fun though.
It's
It's
great.
so much
It's
fun.
like, yeah, I see the glimmer in your eye.
Yeah, it's so much fun.
It is. That's cool. It brings me back to a quote from an earlier episode from Russell Grant, and he said, "Every sample is a person.
Yes,
Every tube is a question asking, am I okay?"
absolutely.
And it presses on my heart every time I think about that, because it really is.
It is.
Because we've if we haven't been there ourselves, we've had a loved one, know somebody who's been in that situation where what's happening?
Yeah.
What is this, and am I going to be okay?
Yeah.
Kind of thing. Yeah.
So I think if you take the entire, disease populations, we're working in pharmaceutical. So I think we should hold that true for every, just because our studies are small doesn't mean anything. We need to hold that same respect for all the samples we collect. I think that's something that I'm much more cognizant now
Right.
in this area. But I think, even oncology, immunology, any sample is so precious. I think we need to think of that way.
Sure.
Yeah.
So you have now this breadth of experience. You've gone from big pharma to small biotech. What lessons have you learned in big pharma that have helped you now, and what vice versa? What lessons can large pharma learn from your experiences in a smaller company?
Yeah. So I say this, I used to say this when I worked for Procter & Gamble and moved to Amgen.
Yeah.
Amgen those days, not Amgen today. It was
Right.
a smaller company.
Right.
So I went from this total quality management system, total quality processes, to Amgen where there were no processes. You were building as we went along, right? So
Clearly,
that was a huge lesson, right? That was a huge lesson. But that entrepreneurial spirit of Amgen, it was so amazing. It was so invigorating.
Yeah.
So I think that's where I'm seeing that now, again, transitioning from Briscoe to Ultragenyx. Take those processes, well-oiled machine of doing something, and we are going into what we call, the hedging at Ultragenyx.
Right.
What can we do? Be entrepreneurial. What's path number one? What is your hedge strategy? So it's like we call it a dynamic drug development. Emil likes to call it the dynamic drug development model, which is really true, because what do we do? How do we pivot from what we learn? So
Right.
that's so important for us in where we are. So I think that, again, could be translated back to pharma. I think if they can learn how to pivot soon, how to stop a trial quickly without wasting the money or processes, that's that. But then what I'm trying to bring into Ultragenyx is not tie down my hands or my lab. We are a small group with all the processes in the world, but what are the essential things that we need to do and be streamlining this?
Right.
Because, we work in all modalities, right? So it doesn't matter which modality, but are there some processes that we can streamline to get the work done.
Sure.
I think that's the big pharma processes. Again, not to tie ourselves down because we want to be nimble.
Right. It's hard to find the right balance.
Yeah. That's
sometimes you have to flex in one direction or another in order to get things done. Yeah.
Yeah.
So do you feel like maybe a greater urgency to fail faster because some things just aren't going to work, and there's some trial and error in this process?
Oh, absolutely.
Yeah.
Absolutely. Fail fast, recover, and move on.
Yeah.
Especially, now that we are looking at all sorts of biomarkers, right? So it's getting harder and harder to understand what these biomarkers mean. But if you don't see anything coming out early on, just move on, get what you can and move on.
Right. All right. Good. As a past president of AAPS and someone who's, passionate about bridging that science, right, and business, what advice would you give scientists who are trying to leap into some of these broader leadership roles?
Be curious.
Okay.
Be curious, because I think, especially when I talk to some of the graduate students at the mentoring breakfasts and all that, it's a lot of linear thinking. I don't know if it is they don't have the passion or what it is.
Right.
Because you have to know what your adjacencies are.
Yeah.
Right? And then that's how you grow. So let's take me as a bioanalytical scientist, right? If I'm always looking at my data, I have all my QCs passing, I'm accurate, I'm precise in the method and everything, and I hand over my data to the PK scientist. Right?
Yeah.
And they plot the graph and they determine dose, they determine half-life, everything. But if I don't have an understanding of what they're doing with my data, what am I learning? Nothing.
Yeah. Right.
I'm not growing.
Yeah.
Right?
Yeah.
So I think that's a piece
That's important.
That's important. Know what your adjacencies are. I call them adjacencies. It's just broadening the knowledge base. And from there on, you have to learn, okay, so this is what they're doing. Okay, so what happens, to this drug? Where is it going? What's the metabolism? What's the distribution? So if you don't keep that curiosity, you're not going to grow. You're going to be linear.
It makes sense.
And I really wish, I was actually thinking, for example, today I was at a session that was a little controversial, but unlike in the past, no one was jumping up and going to the mic. Jolene and I were just talking about that right now. It's like, where is that? There is that passion. We argued right and left at APA.
It was, gloves were off.
Yep. It was so good. And you learned a lot, right? So, so I think curiosity is number one. And number two is the community. This is a community.
Yes.
Take advantage of that community. There's so much knowledge and experience, even around here, right, today at NBC. But so, I think those two is important.
Yeah, I think you make a great point. And when you said be curious, it reminded me of that famous Walt Whitman quote, be curious, not judgmental. Right. And then, take advantage of, there is such a wealth of knowledge, and some people just don't ask, and you're kind of scratching your head. Why not? You have a live captive audience, you're not going to be face to face with this person maybe for another year.
Right.
Exactly.
So exactly.
Good.
Yeah. And then it's like, I feel like, again, all credit goes to people like Ron and Jean and Jeff and all those people who were there, who brought us along. They were ahead in our careers and brought us along. And we are ready to take some people along with us, right? Take advantage of us.
Right. Yes.
Right?
Why not?
Look at Russ. Russ is like Russ Wiener, Russ Grant, all of them. They're opening their arms to educate our younger people.
Yeah. And they're very passionate about that, as are you. Yeah. And it's an interesting, and I kind of wonder how much technology is affecting younger people's desire and ability and courage to take advantage of those face-to-face interactions.
Well, you bring up a good point.
Right?
Yeah.
I think there's something there. You've seen the people that those young folks, they're just interactive.
Yeah.
And they can't wait to, get a chance to reach out and introduce themselves. But there's so many kids that are quiet. You go to any college campus these days, what do you see? Do you see kids walking and talking? Not as much. No, you see people with earbuds on,
Yeah.
looking at the device.
Looking at their devices, yeah, and you don't even.
So-
I mean, even our kids, even my own kids, right? I can. They're upstairs sometimes. I'm texting, "Come to dinner," right?
Right. Texting in the house. Yeah.
That's our life these days. So there may be something there too. But anyway, okay, even if that's the case, right? I mean, they can take advantage of all the AAPS offerings, or doesn't matter. SGCT has all these offerings, and then they can reach out and talk to, then email or do whatever and
Right.
talk to the authors. We can do that too.
Yeah.
So
And I think it's such a collaborative community that they're willing.
oh yes,
They're willing to do that. And some people may be really, really busy, but that's a good question. I'm going to help this young person, and answer it. And, what can we do to help push the science a little further?
absolutely.
And there's that desire.
Yeah, it is the desire for that. Yeah.
Yeah. Awesome. Well,
Yeah.
Chad's going to ask you some questions now.
Okay.
So thank you for spending some time with me.
No, the pleasure to be here talking to you. I feel so
well,
fortunate. Yeah.
It's great. Thank you for giving me the opportunity.
I once did an interview. It's got to go back 10 years, maybe 15 years. I did an interview at a conference with someone, and the question they asked was, "Who's your bioanalytical hero?"
Yeah, you heard me. I have a whole series, right?
What's that? Oh no, I haven't seen that, but I named you.
Oh, you
I
named
named
me.
you
Oh my
and
goodness.
Jean Lee. You and Jean Lee, for real. And it's kind of cool because as I was listening to your discussion with Gregory, I was thinking, wow, yeah, I mean, and you're kind of putting Jean Lee very deservedly, it seems, on some of that pedestal, and I love that. So what I want to do is take some of your advice from when you were talking to Gregory and take advantage of some of your experience and knowledge and ask you a couple things. So one of the areas that's really fascinating, not surprisingly, we talked with Russ quite a bit about rare disease research and how we can make it more effective, how we can partner through some of the challenges in whether it's through discussions at AAPS or through CRO, pharma relationships or what. So what do you think are some of those? Describe some of the challenges and some of your thoughts on what we are doing, what we can do to overcome that.
So I think the sample challenge has to be met.
Mhm.
I think we have a great opportunity to improve on DBS, dried blood spots. So I think that's, if we can get, and then the other, so I'm going to back off a little bit here. The other thing that we all have to learn, the pharma industry, the pharma biotech, is to learn from the diagnostics. Again, this is an age-old issue that we've talked about, right? A long time ago, like that we don't talk, we don't share our knowledge.
Yeah.
Because if we can understand how do they do their newborn screen, right? It's the filter paper, it's dried blood spots. Why can't we capitalize on that to do our analysis, right? I think that's a huge thing that I'm seeing that could benefit that conversation between the diagnostic industry and us. And then as we, so in some cases there are biomarkers that are common to multiple diseases.
Right.
Can those be made into well-characterized diagnostic assays, right? Can that be? Because then we can use one platform or couple of platforms to really generate this data and gather that magnitude of data to see what are the trends in this disease, or do you see that in another disease? So really understanding trends. So that's maybe a data mining thing, or can we develop assays that go into a diagnostic, some kind of a device, and then we can get it out and kind of do the data. So that couple of avenues there, right? How do we get into making it a bit more mainstream diagnostic, and then cure the data, and or get the data from the diagnostics that's currently running, and then gather the data and feed back into our drug development processes. So a couple of avenues there. And then the other piece I think there needs to be is CRO company partnership. One of the challenges that we have, we are a small lab, right? So we are a tiny lab, and we depend on the CROs. But unfortunately, sometimes our sample sizes are so small.
Yeah.
Right?
Yeah.
We don't get the top place in the CRO. We are waiting for 3, 4, 5, 6 months to get in. And then, we will, it gets expensive to us too, right?
Yeah.
Because we are waiting, and then we are at the bottom of the list, as well as, getting our little samples done. It's probably 200, 300 samples compared to thousands.
Or fewer.
Or fewer, sometimes very fewer. Yeah. So these are so the CRO company partnerships matter quite a bit. And then the other piece is also some of these are novel therapeutics, RNA, gene therapy, and cell therapy. So what are some of the challenges we face in those modalities? And then tie it down to, again, I go back to the sample size, go into the sample size, but how can we also, accelerate some of these understanding of those new modalities?
Right.
So those are some of the things that come to my mind.
Yeah. It's just such an important space, but we lump rare diseases together as if they're almost as if it's one space. But rare diseases, it's across all different types of treatments and modalities. But it's so precious. I've seen a few people that have recently moved into going towards rare disease research because of the passion for that. You and Russ certainly come to mind that have just gone in that space. And so that gives me a little hope that just because the recognition that it's so important, that it will go somewhere. So
7,000. That's what I heard. 7,000 rare diseases.
7,000 rare diseases,
and we haven't even scratched the surface.
And it's probably, this is a terrible comparison, I shouldn't even say, but it reminds me of discovering like new species or new like when you hear, they know of 7,000 species of insects in the Amazon, but there's probably 70,000, right? There's probably, or 700,000 rare diseases. And if you could look at the multi genetic variations on those rare diseases, do you think newborn screening needs to be expanded to help?
They're trying to. I don't know too much about it, but they're really trying to put into the newborn screens and just to see how things are. I think there's a process that you have to follow. I'm not well versed in that, but there's a process that you have to follow to get into a newborn screen. I guess. I don't know what it's even called, a menu, whatever. I don't know that.
I'm sure it's very complicated, and you're probably not getting any less complicated these days.
And I think it's statewide, if I'm not mistaken.
It is. Okay.
It is. Each state has different parameters and newborn screen.
Okay, because I remember some years ago when speaking with Don Chase, who was involved in that early research and some of the things that he did in that space, talking about the challenges and how the newborn screening kind of came to be. And so do you think we were talking about the dried blood spots a minute ago? Do you think is it dried blood spots, or is it just the different sorts of more patient-centric sampling, or is there a reason why, or were you just kind of using that as a generic term?
I was using it as a generic term also, but at the same time, the convenience, being able to ship the samples worldwide, right? I mean, so we may be having a patient somewhere far away, and we need to get it shipped to one of our labs. Either in the US or in Europe. And then that's easier to ship.
Right.
You don't have to worry about cold chain.
Yeah. Because it seemed, for a long time, it felt like dried blood spots came into bioanalysis, and it was kind of the tail wagging the dog, right?
Yeah.
Hey, we've got this great solution for a problem that they don't know they have yet in clinical trials.
Exactly.
Have you seen that? Do you think that's shifted? I feel like that's shifted, but I'm not in there in the space you are designing those trials with the.
Yeah, but I mean, I think we also stopped research on DBS. I think there was a time. I mean, it was Mark led a lot of that work too, at Bristol. I think LCMS methods were much more prevalent with DBS than LBAs, but we can do LBAs. I mean, we've done LBAs with dried blood spots. And so I don't know what happened. There was this big hype. We would talk about it all the time in the conferences, and it kind of died down. I don't know if people didn't need it, or that'll be an interesting case study to see what happened. But to me, more and more, now that I'm in the rare disease space, I feel more and more passionate about preserving these precious samples. And sometimes it may not be dried blood spots, because we sometimes need CSF. So that we'll have to see how we get that. But at least to search for biomarkers, I think DBS should be a good place to start for us.
Okay. So you've given a lot of your extra time, your mentor time through APS, right? Past president and involved, building the bioanalytical community. You talked about the arguments. I remember 15, 20 years ago seeing you speak and going, these ligand binding people, yes, they don't think the rules apply to them, right? So I don't even know what question I had now, but I wanted to bring that up. But you've given a lot of your time to APS through the years. Why is that, and what have you gotten out of it?
This was where I grew up. Right, as a graduate student, again, I had great mentors. Dr. Sulanti. She was. It used to be the APQ section,
right?
And she was a abstract screener, right? One day she said, "You're screening abstracts for me." I'm like, "What?" So that's what she did, right? She kind of encouraged us to participate as graduate students. And the next thing I know, I'm like screening chair and all that. And so I started the ground level, screening abstracts. And then I grew up with APS, and I've seen the growth of APS and where it is today. So proud of the org, and I want to give back to APS.
That's phenomenal. That's phenomenal. So I want to get one other thing on record, because you've told me the story before about how you came from Sri Lanka to KU. So if you wouldn't mind telling us that an abbreviated maybe version of that story, because I think it's really I think it's really cool.
So I grew up in Sri Lanka. Did my formal education and also undergraduate in analytical chemistry. And then, so I was very fortunate to be interviewed by Dr. Neely. He was in Oklahoma, I believe, at that time. There was a 13-university consortium that did interviews in Southeast Asia to recruit graduate students. So, and I got interviewed, and they represented some of these universities. So I got into Kansas. I came to Kansas to work for Dr. Wilson, George Wilson, who was, like I said earlier to Greg, he was an electrochemist. But I think that was the foundation, KU, as well as Dr. Wilson's vision to say, there's nothing to go be pure chemistry anymore.
Right.
And to tell us to go learn a lot of the other.
I love it.
Other disciplines, and then finishing my graduate degree in bioanalytical. Where, those days we had to go to the farm in Eudora to bleed the goats, and you did it all, right? You did it all, made your own antibodies.
Bleed the goats.
And had a hybridoma facility. Faced the consequences of humid summers in Kansas with, contamination. So we had to do it all, right, from scratch. And, getting the degree there. And then being fortunate, being in KU, being fortunate enough to have pharmaceutical chemistry department next door, right? And having that influence of the pharmaceutical scientists coming and talking to us, giving us seminars. Even we had a course where the FDA came and gave us this whole course on what IND, GLP, ND, those were like alphabet soup at that time. It went over my head. But then you realize, oh my gosh, you learned this in school, now you're applying it in industry. And then so I was fortunate. I was able to do a postdoc in pharm chem,
Right.
drug conjugate transport to solid tumors. So getting that breadth of knowledge.
Yeah.
So that's kind of my story.
That's phenomenal.
And I'm going to give back to KU because Dr. Sulanti, she's going to Sri Lanka
Oh, okay.
to recruit recruit students.
She is. And you're going with her?
I'm going with her.
Oh, you better. Yeah. Yeah.
Oh my God, we're going to have a blast. So
Wow.
Sue will go to two of the main universities in Sri Lanka, but we will get students from everywhere so that Sue can interview. But I also want to show Sue our country. It's a beautiful country, so I'm going to take her on a little tour for about a week.
Amazing.
So I'm so pumped.
When are you going?
July.
In July.
July.
Amazing.
Yeah. So I'm ready to get back to KU in that way.
Yeah.
Yeah, because I benefited, and so some other Sri Lankan students may benefit coming to Kansas.
Yeah. Well, I benefited by knowing you.
Oh, thank you. I benefited from all of you.
Thank you so much, and thanks for taking the time to talk to us.
Yeah, thank you very much. So this was fun.
Yeah, it was.
All right. Thank you. Thank you.
Chad and I would like to extend a very warm thank you to AAPS for all the wonderful support, allowing us to be here and sort of take over the media room for a while to have some wonderful conversations with some friends and some new friends and scientists here at AAPS NBC. A special thank you to Rebecca Stouffer, who has been phenomenal in her support and helping all this happen very smoothly. So we want to thank her, thank the organization, and we look forward to seeing you soon.